So dear students, today we are going to discuss about control of gene expression in prokaryotes. So the regulation of gene expression is very critical for all organisms because sometimes some genes uh, may be switched off, sometimes some genes may be switched on, depending upon the requirement of a cell. So there are a lot of factors and elements which can control, regulate the expression of genes in all organisms. They fall under three categories. They are genes and regulatory elements, levels of gene regulations, and DNA binding proteins. So first of all, genes and regulatory elements. There are different kinds of genes and gene product which can regulate the gene expression. First of all, the structural genes that encode structural proteins. And the second one is regulatory genes that encodes product that interact with other sequence and affect their transcription and translation. And there are additionally regulatory elements that are DNA sequence that are not transcribed but play a vital role in regulating gene expression. So these are the three major categories that will regulate gene expression. Regulatory genes actually codes for proteins that will interact with other genes to affect their switch on and switch off. And regulatory elements, they may not be the part of the gene system, but they may not be transcribed, but their role is very critical in regulation of this gene system. Gene expression may be constitutive, that is, they will be continuously expressing under normal cellular conditions. There is positive control. This means it will stimulate the expression of gene. So it will enhance the expression of gene. That is positive control. The next and the last one is negative control. Negative control means it will inhibit or interfere the gene expression. So it will try to switch off gene transcription and translation. In positive control, it will accelerate or enhance the activity of gene. So these are the levels of gene expression and control by regulatory elements or regulatory genes. So here we are going to discuss about the levels of gene regulation. There are different regions at which regulation can be possible. So first of all, if you consider a compact DNA, before it get transcribed, it will relax by some enzymes. Then it will undergo transcription, and the transcribed product is a pre-messenger RNA. Pre means it is not a processed one. There are a lot of regions which should be cut before it will form before it becomes a mature mRNA. So this is known as mRNA processing. So this processed mRNA, that means a lot of post-transcriptional modification will occur in pre-messenger RNA. So this processed mature messenger RNA will undergo translation, and the translated product will be an inactive protein, and it will undergo post-translational modification to become a modified active protein. So there are sites at which regulations can be possible. So mRNA processing is one step. Post-translational modification is also there. So there are different sites at which gene regulations can be controlled. So there are number of binding proteins, DNA binding protein, that can regulate gene expression. They are domains. Domains means 60 to 90 amino acids responsible for binding to DNA, forming hydrogen bond with the DNA. So here you can expect a direct interaction between DNA and the domain. Second one is motif. They lies within the binding domain, a simple structure that fit into the major groove of DNA. DNAയുടെ മേജർ ഗ്രൂവിലേക്ക് കറക്റ്റായിട്ട് ഫിറ്റ് ചെയ്യുന്ന ബൈൻഡിങ് ഡൊമൈനിൽ തന്നെയുള്ള ഒരു സ്ട്രക്ചറാണ്, സിമ്പിൾ സ്ട്രക്ചറിനെയാണ് മോട്ടിഫ് എന്ന് പറയുന്നത്. Different types of motifs can be seen. So here you can see many type of motifs. Helix turn helix is there, Singer finger motifs are there, leucine zipper motifs are there. So first of all, helix turn helix, it got its name from its structure. Here you can see that a helical structure is there, and intermediate and the middle region you can see a turn which connects to helix. So the shape of the protein is like a helix turn helix. So that's why they got this name. So here you can see DNA binding helix, turn, dimer binding helix. So the next one is zinc fingers. Zinc ions will be there. So here the structure is like zinc plus fingers. Leucine zippers are there. Here amino acid, a particular amino acid will be there. It is leucine, and leucine zippers are there. These are the common DNA binding motifs. As we told earlier that helix turn helix can be seen in both prokaryotes and eukaryotes, and their characteristics include 2 alpha helices and 1 turn will be there, and their major binding site in DNA is major groove. Then again, zinc fingers, eukaryotic regulatory and other proteins. Characteristic features includes loop of amino acids with zinc at base. They binds with major groove. Steroid receptors are there. Leucine zippers, eukaryotic transcription factor, helix of leucine residues, and basic arm. 2 leucine residues interdigitate. They will bind with 2 adjacent major grooves. So helix, loop helix is there, homeodomain is there. So this is actually the representation of all available DNA binding motifs which works in prokaryote as well as eukaryotes, and their major binding regions in DNA is also represented. So now we are going to discuss about gene regulation in prokaryotes. That means Operon system that controls transcription in bacteria. So in Operon structure, we will discuss about the Operon structure, negative and positive control, inducible and repressible operons. We are going to discuss about the details of lac operon in E. coli, lac mutations, and positive and catabolic repression, and the last one is TRP operon. So these are the topics which we are going to discuss in details under transcriptional control in bacteria or in prokaryote. So before we start, what is an operon? Operon is a gene system which consists of many genes, and together they will produce a product, and that will regulate the expression of a gene. So an operon consists of a promoter region, an operator region, and many structural genes. In addition to that, there are regulatory genes that are not a part of operon but can control the expression of genes. Okay? So operon എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ ഒരു പ്രൊമോട്ടർ റീജിയൻ ഉണ്ടായിരിക്കും, ഒരു ഓപ്പറേറ്റർ റീജിയൻസ് ഉണ്ടായിരിക്കും, പ്ലസ് സ്ട്രക്ചറൽ ജീൻസ് ഉണ്ടായിരിക്കും. കൂടാതെ ഇതിൻ്റെ പാർട്ടല്ലാതെ, ഡയറക്റ്റ് പാർട്ടല്ലാതെ വേറെ ഒരു ടൈപ്പ് ഓഫ് ജീൻസുകളുണ്ട്, റെഗുലേറ്റർ ജീൻസുകളുണ്ട്. ദാറ്റ് ജീൻസ് ആക്ച്വലി പ്രൊഡ്യൂസ് എ പ്രോട്ടീൻ പ്രോഡക്റ്റ് ദാറ്റ് മേ റിപ്രസ് ഓർ induce the expression of structural genes in an operon system. So ഇതാണ് നമ്മൾ മനസ്സിലാക്കേണ്ടത്. ഒരു operon system എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ ഒരു ഒരേ ടൈമിൽ പ്രവർത്തിക്കുന്ന കുറച്ച് ഓപ്പറേറ്ററും പ്രൊമോട്ടറും സ്ട്രക്ചറൽ ജീൻസും എല്ലാം കൂടി ചേർന്നിട്ടുള്ള ഒരു ജനറ്റിക് സിസ്റ്റം ഇൻ പ്രോകാരിയോട്ടസ് operon. അതാണ് മനസ്സിലാക്കേണ്ടത്. So from this picture you can see that how an operon works. First of all, you can see a regulator region on the left side. It has its own promoter. It will undergo transcription and translation and produce a regulator protein. Okay, this is actually not a part of operon system, but they can play a vital role in controlling gene expression of structural genes. On the right side you can see that promoter region is there where RNA polymerase will bind, and just next to promoter region is operator region, where these are the possible sites where the product of regulator gene will binds. So promoter region is followed by operator region, where you can expect the binding of regulator protein. Then comes the genes, structural genes, gene A, B, C. So how it works? നമ്മളിവിടെ നോക്കുകയാണെങ്കിൽ റെഗുലേറ്ററിന് ഒരു പ്രൊമോട്ടർ റീജിയൻ ഉണ്ട്. അവിടെ വന്നിട്ട് അതിൻ്റെ RNA പോളിമറൈസ് വന്ന് ബൈൻഡ് ചെയ്യുകയും റെഗുലേറ്റർ ജീന് അണ്ടർഗോ ട്രാൻസ്ക്രിപ്ഷൻ ആൻഡ് ട്രാൻസ്ലേഷൻ, ഇറ്റ് വിൽ പ്രൊഡ്യൂസ് എ റെഗുലേറ്റർ പ്രോട്ടീൻ. അപ്പോൾ റെഗുലേറ്റർ പ്രോട്ടീൻ എന്ന് പറയുന്ന റെഗുലേറ്റർ എലമെൻറ്റ്സ് ദാറ്റ് ആർ നോട്ട് എ പാർട്ട് ഓഫ് ദിസ് ഓപ്പറോൺ സിസ്റ്റം. ഇനി ഇവരെന്ത് ചെയ്യും? നമ്മൾക്ക് ഒരു പ്രൊമോട്ടർ റീജിയൻ ഉണ്ട്. ആ പ്രൊമോട്ടർ റീജിയനിലേക്ക് RNA പോളിമറൈസ് വന്ന് ബൈൻഡ് ചെയ്യുന്നു. ബൈൻഡ് ചെയ്തതിന് ശേഷം ഇഫ് ബൈൻഡിങ് ഇസ് ഓക്കെ, then primary recognitions are over, then RNA polymerase എന്ത് ചെയ്യും? will moves through operator region. operator regionൽ regulator proteins enhance ചെയ്യുന്നതാണെങ്കിൽ, അതല്ലെങ്കിൽ ഇൻഡ്യൂസ് ചെയ്യിപ്പിക്കുന്ന ഒരു പ്രോട്ടീനാണ് റെഗുലേറ്റർ പ്രോട്ടീൻ എങ്കിൽ RNA പോളിമറൈസ് ഈസി ആയിട്ട് ഓപ്പറേറ്റർ റീജിയനിലൂടെ കടന്നു പോവുകയും സ്ട്രക്ചറൽ ജീൻസ് ആയിട്ടുള്ള എനെയും ബിനെയും സിനെയും ട്രാൻസ്ക്രൈബ് ചെയ്യുകയും ചെയ്യും. അതുവഴി ട്രാൻസ്ലേഷൻ പ്രോഡക്ട്സുകളും ഉണ്ടാവും. ഇനി ഓപ്പറേറ്റർ റീജിയനിൽ വന്ന് ബൈൻഡ് ചെയ്യുന്ന റെഗുലേറ്റർ പ്രോട്ടീൻ റിപ്രസിബിൾ ആണെങ്കിൽ, അഥവാ ഈ ഒരു ഓപ്പറോൺ സിസ്റ്റത്തിനെ സ്വിച്ച് ഓഫ് ചെയ്യേണ്ട ഒരു പ്രോട്ടീൻ പ്രോഡക്റ്റ് ആണെങ്കിൽ RNA പോളിമറൈസിന് എന്ത് ചെയ്യാൻ പറ്റില്ല? ഓപ്പറേറ്റർ റീജിയൻ കടന്നുപോയി ജീൻ എ ബി സിനെ ട്രാൻസ്ക്രൈബ് ചെയ്യാൻ പറ്റില്ല. സോ എന്ത് സംഭവിക്കും? ട്രാൻസ്ക്രിപ്ഷൻ will be off. as I told you earlier that there are negative and positive control, inducible and repressible operons. What do you mean by inducible operons? Actually, inducible means they must be induced. Actually, they are in a off state and needs to be on. So this operon will be transcriptionally off. So it must be induced to be turned on. So this is known as inducible operon. Next one is repressible operon. They are actually transcriptionally active and they are in an on state and needs to be off. This is known as repressible operon. അപ്പോൾ ഫസ്റ്റ് ഓഫ് ഓൾ ഇൻഡ്യൂസിബിൾ മീൻസ് അത് ഓൾറെഡി ഓഫായിട്ട് കിടന്നുകൊണ്ടിരിക്കുകയാണ്, അതിനെ ഓണാക്കണം, ഇൻഡ്യൂസ് ചെയ്ത് അതിനെ ഓണാക്കണം. റിപ്രസിബിൾ മീൻസ് ഇറ്റ്സ് ആക്ച്വലി ഇൻ ആൻ ഓൺ സ്റ്റേറ്റ്. അപ്പോൾ ഓണായിക്കൊണ്ടേയിരിക്കുകയാണ്, അതിൻ്റെ പ്രോഡക്റ്റുകൾ എക്സസ് ആയിട്ടുണ്ടായിട്ടുണ്ട്, ദെൻ ഇറ്റ് മസ്റ്റ് ബി ടേൺഡ് ഓഫ്. കാരണം പ്രോഡക്റ്റ് ഒരുപാട് ഉണ്ടായിക്കഴിഞ്ഞ് കഴിഞ്ഞാൽ ദർ ആർ ലോട്ട് ഓഫ് റിപ്രഷൻ മെക്കാനിസംസ് ആർ ദർ. അപ്പോൾ അത് കാരണം ഫീഡ്ബാക്ക് ഇൻഹിബിഷൻ അങ്ങനത്തെ കുറേ കാര്യങ്ങൾ നിങ്ങൾ പഠിച്ചിട്ടുണ്ടാവും. അപ്പോൾ അങ്ങനത്തെ ഒരു പ്രശ്നം കാരണം ഓവർ പ്രോഡക്റ്റ് ഉണ്ടാവേണ്ട ആവശ്യമില്ല. അപ്പോൾ അതുകൊണ്ട് തന്നെ ആ ഓണായിക്കൊണ്ടിരിക്കുന്ന ജീൻ സിസ്റ്റത്തിനെ ഓഫ് ആക്കണം. അപ്പോൾ ദിസ് ആർ ടു ടേംസ് വിച്ച് ആർ വെരി ഇമ്പോർട്ടൻറ്റ് ഇൻ പ്രോട്ടീൻ ജീൻ റെഗുലേഷൻ ഓഫ് prokaryote: inducible operon and repressible operons. So the next term you have to study is negative and positive control. Negative means the product of regulator gene will be an inhibitor. So always negative means that the product of regulation is an inhibitory protein that will try to inhibit gene expression. So in negative inducible operons means the control at the operator site is negative. Molecule binding to the operator inhibiting transcription. Such operons are usually off and need to be turned on so the transcription is inducible. Here a molecule that will try to induce gene expression is known as inducer. So negative inducible operon means ഓപ്പറേറ്ററിൽ നിന്നുള്ള ആ ഒരു കൺട്രോൾ എന്ന് പറയുന്നത് നെഗറ്റീവായിട്ടുള്ള ഒരു എക്സ്പ്രഷനാണ്. അങ്ങനെയുള്ള ഒരു സാധനം ഇൻഡ്യൂസിബിൾ ആണ്. നെക്സ്റ്റ് ടേം എന്ന് പറയുന്നത് ഇൻഡ്യൂസിബിൾ എന്നാണ്. അഥവാ നോർമലി അത് ഓഫാണ്, അതിനെ ഓൺ ആക്കുകയും വേണം. നെഗറ്റീവാണ്, control level at operator site is negative and the operon system must be on. അപ്പോൾ ഓൺ ആക്കണം എന്നുണ്ടെങ്കിൽ ഒരു ഇൻഡ്യൂസർ എന്ന് പറഞ്ഞിട്ടുള്ളൊരു സ്മോൾ മോളിക്യൂൾ ഉണ്ട്. ആ സ്മോൾ മോളിക്യൂളാണ് ഈ സാധനത്തിനെ സിസ്റ്റത്തിനെ ഓൺ ആക്കുന്നത്. അതിനെയാണ് ഇൻഡ്യൂസർ എന്ന് അറിയപ്പെടുന്നത്. So here you can see how it works. First of all, no inducer is present. So on the left side you can see regulator will produce a will undergo transcription and translation and will produce a protein product that is an active regulator protein. This active regulator protein binds with promoter region. So the product of regulator gene here is a repressor that will repress this whole genetic system. So what happens to RNA polymerase? RNA polymerase will binds with promoter region and they will try to pass operator region, but there is an obstacle. The active regulator protein is there and that active protein is also a repressor. So RNA polymerase cannot bypass this region. So what will happen? Structural genes will not be transcribed. So the next term, the inducer. If inducer is present, what will happen? The same as the scenario. On the left side, a regulator protein will produce an active repressor molecule. Because this operon system is negative. Negative means regulator protein will produce a protein product that will be a repressor. Negative means repressor. Negative repressor. Inducible means it's actually off. It must be on. So if an inducer is present, then transcription and translation of regulator protein will produce active regulator protein that will not bind with operator region, because the inducer molecule acting as an inducer, when inducer is present, it binds with the regulator protein and thereby making the regulator unable to bind with operator region. So this is how an inducer works. So what inducer does is, it will binds with the transcriptional regulation protein, and by which it cannot bind with operator region. So RNA polymerase will recognize the promoter sites of this operon system, and it will goes on and will pass operator region, will cross operator region, then will undergo transcription and translation of structural genes. Transcription and translation will occur. So this is how an inducer works. So actually inducer is a molecule that binds with a regulator repressor protein and make them not able to interact with the promoter region. So this is how an inducer works. So in the previous slide, we had discussed about negative inducible operons. That means regulator protein product will be a repressor. That is negative. Repressible means it is actually in an on state and it must be get off. So this is negative repressible operon. The control at the operator site is negative because the product of regulator gene is a negative or a repressor. But such transcription is usually on and needs to be off. So the transcription is repressible. So how it works? Here, like inducer, there is another molecule which is known as co-repressor, a small molecule that binds to the repressor and makes it capable of binding to the operator to turn off transcription. So this is just the opposite of inducer. It will binds with the repressor and makes them capable of binding to the operator to turn off transcription. But transcription off ചെയ്യാൻ വേണ്ടി റിപ്രസറിനെ ഒന്നുകൂടെ ഉഷാറാക്കുന്ന ഫംഗ്ഷനാണ് കോ റിപ്രസർ ചെയ്യുന്നത്. സോ നെഗറ്റീവ് റിപ്രസിബിൾ ഓപ്പറോൺ വാട്ട് കോ റിപ്രസർ ഡസ് ഇസ് ഇറ്റ് വിൽ എൻഹാൻസ് ദ ആക്ടിവിറ്റി ഓഫ് റിപ്രസർ. So now we are going to discuss about negative repressible operon. As the name indicates, negative means the product of regulator will be a repressor. And repressible means the operon is usually in a turned on condition and it must be turned off. So this is the overall thing you need to know. Negative repressible operon. So in the first condition, No product U, here U is a co-repressor. No product U is present. That means co-repressor is absent. So what will happen? Regulator protein will transcribe and translate and produce inactive regulator protein. Inactive regulator protein, that is, here the repressor is inactive. So inactive regulator protein cannot bind to promoter region, operator region. So transcription and translation of structural genes will occur that will produce a product U. So in the second case, when the product U is present, here also transcription and translation of regulator protein will occur. This will result in the formation of inactive regulator protein. Here it is the repressor, and it will bind with regulator protein. The product U, here it is a co-repressor and makes the inactive regulator protein to an active one. So the regulator protein now can bind to the operator region and prevent RNA polymerase transcribing structural genes. So here how the system works. When product U is present, then inactive repressor becomes active and it can turn off transcription and translation. So here on the right side of part A, case A, you can see a biochemical pathway by which precursors T, intermediate product, and product U will be produced. This product U can act as co-repressor in the second case, in the B part of this negative repressible operon. So this is all about negative repressible operon. നമ്മൾ മനസ്സിലാക്കേണ്ട കാര്യം, negative means regulator protein will be repressor. Repressible means usually on. അതിനെ ഓഫ് ആക്കണം, റിപ്രസ് ചെയ്യണം. അതാണ് നമ്മൾ മനസ്സിലാക്കേണ്ടത്. സോ കോ-റിപ്രസറും ഇൻഡ്യൂസറിൻ്റെയും ഫങ്ഷൻസ് നമ്മൾ ഓൾറെഡി ഡിസ്കസ് ചെയ്ത് കഴിഞ്ഞു. ദിസ് ഈസ് ഓൾ അബൌട്ട് നെഗറ്റീവ് റിപ്രസിബിൾ ഓപ്പറോൺ. So the next section is inducible and repressible operons. Here another term you have to be familiarized with is positive inducible operon and positive repressible operon. Now you will be able to understand what will be positive inducible operon. Positive means the regulator protein or regulator product will be an inducer. Inducible means it is usually in an off condition and it must be turned on. So positive inducible operon means regulator protein will induce transcription and translation of this inducible operon. Next one is positive repressible operon. That means here also the gene product will be a positive or enhancer, or we can call that it is an inducer. It can enhance the activity of, it can promote the activity of structural gene transcription. So repressible means it is usually in an on condition and it must be turned off. So what will happen to this operon? We will discuss it later. So here is the conclusion of all operons we had studied. First one is negative inducible. Negative means the regulator protein will be a repressor. Here it is an active repressor, so it will binds to the operator region and prevents RNA polymerase, so transcription will be off. When the product or substrate makes the repressor inactive, so if a product is present, then it will binds to the regulator protein. So regulator protein will be unable to bind to the operator region, so transcription will be on in the presence of the product. The next case is negative repressible. Here also regulator protein will be a repressor. Here it is an inactive repressor, so it cannot bind to the operator region, so RNA polymerase will transcribe and translate. Transcription will be on. In the presence of a substrate, in the presence of a molecule, in the presence of a product makes the repressor active. That means now it is able to bind to the operator region, so it can interfere RNA polymerase transcribing the structural genes, so transcription will be off. In the third case, it is positive inducible. That means here the regulator protein will be an inactive activator, so inactive activator cannot binds to the operator region, so transcription will be off. And now a substrate makes the inactive activator to active, thus it can binds to the operator region and enhance the activity of RNA polymerase, so transcription will be on. In the fourth case, positive repressible means, positive means here the regulator protein will be an activator, but here it is an active activator, so transcription will be on. And product makes the activator inactive, that means transcription will be off. So this is the summary about different types of operons we had just studied.